Anybody have a problem? Are there any questions in the chat box? People just don't know how to even define dysplasia. Okay. And what is the meaning of disorderly growth? And that's where all this story of, and we said it is grades of architectural disorganization. So what do you mean by architectural disorganization? Take squamous epithelium. What? What do you mean by architectural disorganization? We understand atypical cytomorphology, and you can see it. Okay, the cells are larger, there is cytoplasmic, the nuclear cytoplasmic ratio is altered. You also see some abnormal looking nuclei, they show you crinkling, whatever else. Okay, but what do you mean by grades of architectural disorganization? Anybody wants to answer that question? Yes, Anis?
Ma'am, especially if you take it as squamous epithelium, ma'am, the cells are arranged in an orderly manner. They are in layers, ma'am. Actually, if you see here.
I know this is what somebody mentioned, and I think it was Ashrita. She said lack of polarity. Okay. Yes. So
there is haphazard
also
arrangement
polarity. Now see this picture down here. It's there in all. Okay. But you see it very clearly on this left-hand side picture. Okay. All the cells in a normal epithelium should be, the basal layer should be perpendicular to the basement membrane. Yes. And above that, they should flatten. All the cells should flatten, and then they become parallel to the basement membrane. That is normal polarity, because polar means in respect of something. So in respect of the basement membrane, this is how we find normal architectural organization. Now, we said that neoplasia, in one way or the other, and the more severe it is, the more likely it is to be malignant, is you get severe disorganization, architectural disorganization, and now you can see it. Do you think all of these are placed nicely parallelly? Some are, placed at angles, etc., etc. And of course, you see atypical cytomorphology. So this can not be called normal, and this is what is called lack of polarity. Has anybody got any questions on that, please? And if it's in the chat box, please let me know.
Yes, ma'am. This one is poorly differentiated and de-differentiation similar.
Poorly differentiation and de-differentiation is both of them have got poor differentiation, but de-differentiation assumes that once upon a time that tumor had better differentiation, okay, and now that differentiation is lost. It is now losing its differentiation. So say I had pleomorphic adenoma and I knew it looks very benign and everything else, then it de-differentiates to a carcinoma. Or I have a nice, well-differentiated chondroid tumor, which you can see the hyaline cartilage and everything, and then suddenly it goes wild. Now you might ask, why or how does it de-differentiate? Now I don't think that a fully differentiated cell is able to go back. So it is a tumor. There are stem cells in that tumor, and those stem cells are differentiating beautifully, all right, into the fully mature form. And for all practical purposes, that was benign. And then what happens, those stem cells, something happens to them, either they acquire a mutation or there is a change in the environment in which those tumors are growing. And you know that can happen if, for instance, we have a better or less well-differentiated tumor that is given radiation therapy, okay? And that can convert a nice, well-differentiated tumor when it recurs into a very high-grade tumor. So the stem cells there have had something, that a switch in their genome that makes them less susceptible to the differentiating influences that are coming to that tumor. Is that all right? Has somebody understood that? We can discuss it again
Yes,
if you wish.
ma'am. So, as an extension to that, is de-differentiated same as undifferentiated? Is another question.
Yes, it is. No, then it becomes undifferentiated. Of course. But we know that it was well differentiated and therefore we call it de-differentiated. If you never knew that there was a well differentiated tumor, then you call it undifferentiated. It's as simple as that. Okay.
Right. And
so there are some that come that are undifferentiated de novo, and we know those tumors. Some of them we talked about, and you know that we call the anaplastic thyroid carcinomas. We gave that example. Yes, it starts out as being well differentiated, follicular or papillary, goes through a poorly differentiated, and then it becomes totally undifferentiated and anaplastic. And when you see this patient at the last stage, you may not be able to see the original well-differentiated tumor from which it arose. All right? Very
ma'am,
good.
one more question. What is the
Yes.
difference between high-grade dysplasia and carcinoma in situ?
There is none. It's the same thing. Okay? So,
that's all, ma'am.
yeah. All right. Very good. So now this is what this is. Okay? And now we have terminology. And that's what I was asking you. The terminology for dysplasia and carcinoma in situ could be in situ neoplasia, and you can have cervical in situ neoplasia, you can have vulval in situ neoplasia, in which you call it VIN. In the endometrium, you call it EIN, that is endometrial intraepithelial neoplasia. You say, or atypical hyperplasia. Some people call it atypical hyperplasia, okay? And then there is this term EIC, which is endometrial in situ carcinoma. That is used for the serous type of intraepithelial neoplasia, whereas EIN is used for the endometrioid. I don't want to sort of split hairs here, because most of you may not even have heard of serous carcinomas of the endometrium. Okay. Then in the breast, you say atypical ductal hyperplasia and you say duct carcinoma in situ. There is absolutely no histological difference between the two. Okay. If you have low-grade DCIS and ADH, it is only the extent. So if you have a larger extent of tumor, you call it low-grade DCIS, otherwise you call it atypical ductal hyperplasia. So you're getting the idea that pathologists are now splitting hair, okay? And so you have DCIS of low, intermediate, high. And I just told you that adenomas in the colon, in the stomach, etc., are graded with low, moderate, and high-grade dysplasia. So now all my 9 students over here, do you agree that dysplasia is neoplasia? Is it? Do you agree or you don't?
Yes, ma'am.
Yes. So all of you said no, but I'm glad that you have understood this. All right. Now, I'm going to ask, this is now cervix. Okay. So who has done cytology? Because some of you who have done in the first year have not done cytology. So Ashweta, have you done cytology?
Yes, ma'am.
You've done?
Yes, ma'am.
Which
Yes,
year are you in? 18 months you've done?
14, ma'am. I'm in the 14th.
14 months. So did you go through cytology? You
Yes,
see
ma'am.
pap smears? You have?
Yes, ma'am. I have seen.
So maybe you can answer this question. Okay, who's that now? Was it Ashrita or was it Anvesha? Ashrita, yes. All right, tell me. This is CIN, this is normal, okay? This is CIN1, this is CIN2, and this is CIN3. All right,
Yes, ma'am.
is the diagnosis of 1, 2, and 3 possible on a pap smear? The answer is simply yes or no.
No, ma'am.
No, ma'am. All right.
Possible, ma'am.
Anvesha?
Ma'am, possible.
Possible. Okay. Aishwarya?
Ma'am, like counterparts can be given, not the exact diagnosis in 1, 2, and
Fine, but it's the diagnosis of CIN 1, 2, and 3. You'll call it LCIL and HCIL. Doesn't matter. So let me say LCIL. Is the diagnosis of LCIL possible by pap smear?
Yes, ma'am.
Yes, it is. So
Yes,
in
ma'am.
other words, you're telling me that CIN1 can be made, right? The diagnosis?
Yes, ma'am.
Excellent. Sumita, is it possible?
Yes, ma'am.
All right. Tejas?
Yes, ma'am.
Oh, okay. Ujjwal?
Yes, ma'am.
Excellent. So all of you said, and the answer is absolutely right. Has the chat box said it is yes? Yeah, because everybody
Yes,
will
ma'am.
make the diagnosis, right? But now I want to know, you see the difference over here? You know, no, who, all of you will tell me what is CIN1 and how do you differentiate it from CIN2 and from CIN3? Will somebody tell me on histology? And I've got the pictures for you here. How do you differentiate that?
The extent of dysplasia.
This extent of dysplasia, what do you mean by that, Ashrita?
The full thickness, or it is.
Full thickness, okay. That is CIN3, right?
Yes, ma'am.
Excellent. Then
In the
that is
CIN1,
full
it will be
thickness. So that means
less
the
than
whole
1/3.
epithelium. Then, so what about CIN2? Come on, Ashrita.
Ma'am, if it is less than 1/3 of the thickness, we call like SIN1.
SIN1. So here, less than 1/3, it is SIN1 in this big picture. And then
If it is more
2/3,
than one,
it is, which you see over here. It is
it is
CIN2, right?
SIN2.
Excellent. And this. So now, Ashrita, or any one of you, you're going to tell me, how do we make a diagnosis on pap smear? We take a smear, right? So we take one of these swab sticks, or we take a spatula, and then we take the superficial layers, right?
Yes, ma'am.
Yes, and we smear them, correct?
Yeah, ma'am.
But I heard, Ashrita, just now, you telling me that the abnormality is in the lower 1/3 in CIN1, it is 2/3 in CIN2, and it is full thickness in CIN3. So I will be able to make a diagnosis of CIN3, no? Because the top layers are come out on my smear. So how do I make the diagnosis of CIN1 and 2? What do I do? I scrape like that? No, I don't. So come on, Ashrita, how do I do it? Because you all answered yes.
In PAP, we can give like L-seal and H-seal depending upon the
I
other
don't
features.
care whether you call it L-sil or H-sil, or you call it CIN1, 2, which CIN1, which is L-sil, and the other two are there. I want to know if the abnormality is in the lower 1/3 or lower 2/3, how do I make the diagnosis on a cytology smear? Because I'm just taking the superficial cells. Yes or no?
Yes, ma'am.
Oh, very good. So, Anvesha, everybody answered yes, 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 yes. And all the people in the chat box said yes. So now you tell me, I can make the diagnosis of CIN3, that is H-sil.
H-sil.
Because, yeah, there in the top, here I'm scraping away the top, okay? And I'm smearing it, so I can. But now please tell me, how? That's the question I'm asking. The question is how? Yes, Manish.
Ma'am,
That's why I'm showing you know the pictures.
yes,
You
ma'am.
have to imagine.
The based on the presence of the other cytologic
What
cells,
cytological
which
criteria?
which
You tell me, no, the lower 1/3 or the lower 2/3 is abnormal. So then how do I make this diagnosis on a smear which only gets the superficial cells? Anis, you know my question, no?
ma'am,
Ah.
we can have like for CIN1 you'll find mature squamous cells also, no, ma'am? Like
You'll find what?
Along with those atypical cells, you can have
Along with atypical cells. So you're going to see atypical cells which have come from the bottom. You scraped it so hard. Yes, Anis? Anybody there who wants to answer this question? Anybody in the chat box? See, I know that you guys, that's why I am not asking you to imagine, I am asking you to see. What did we say was the diagnosis of dysplasia? Doesn't matter, you call it H-sil, you call it anything. what is the definition of dysplasia? Yes, Anvesha, she's very good at definitions. Yes. What is it?
Intraepithelial neoplasia, ma'am.
Very good. Intraepithelial neoplasia. So, but what is it? What did we define dysplasia as?
Abnormal site.
Disordered
Then-
That's not what we defined it as. Come on, what did we define it as?
Ma'am,
Progressive
progressive
lack of
differentiation.
Did you say?
Epithelium.
It is progressive lack of differentiation in epithelium. Yes? So what is the meaning of progressive lack of differentiation? What is the meaning? Because you are very good, all of you, at learning by heart all definitions. I'm wanting to understand, do you understand? All right? So, you're seeing this, no? Do you see this over here? Now, this is CIN1, yes? And
No.
all of you are telling me that it's the lower 1/3 that is involved. Now, what is the meaning of differentiation or lack of differentiation? Is it only in the lower 1/3? You mean those cells in the lower 1/3 are abnormal and everything above it is absolutely normal? It cannot be, because if you have cells and the stem cells or the parabasal cells that are there at the near the bottom, if they are abnormal, yes, they are obviously going to give rise to progeny, which are also abnormal, yes? And those abnormal cells, if they show you no differentiation at all, they become CIN3. Right. If they show a good deal of differentiation, it means you still get some loss of polarity, you will still get maturation, but that maturation will be arrested at some stage, because it cannot reach the stage of showing you nice superficial cells. So the superficial cells that you see here in the normal will probably get as far as the high intermediates, and those high intermediates will show you the abnormal nuclei. So when Anis was saying, oh, you will see some abnormal cells along with it, what abnormal cells is it that you're seeing? You are seeing, and I will show this to you. Here you can actually see it, that the top layers are not looking like what you should see in normal. They are actually showing you over here in the CIN2, it is showing you abnormal cells at the top. So please don't even think about this ridiculous definition that the lower 1/3 is abnormal, the lower 2/3 are abnormal. What is that abnormality that is in the lower 1/3 and the lower 2/3? Come on, Anis, what is that abnormality? It's an abnormality right throughout the epithelium. But what is that abnormality that all students are talking about when they say lower? What is it?
Pardon, ma'am. Hello.
Yes, Anis.
progressive.
The differentiation that is.
Oh,
Yes,
so
I
it
know.
is the basal layers and the
basal
basal layers. It looks blue because the differentiation is less and less. So it is most over here. Yes, you have a few layers of basal and parabasal cells, and then it starts to differentiate. And you see how they are flattening, but you still see that the nuclei are all not nice and parallel. Some of them are, sort of placed perpendicular, some obliquely, and I'll show you that. And this is also true over here. You have loss of polarity right throughout the epithelium. So please don't believe that it's only lower 1/3, lower 2/3, or full thickness. This full thickness is complete lack of differentiation. Here there is good differentiation, but it is lagging and you have abnormal nuclei. Over here, you have some degree of differentiation, certainly more than CIN1, but certainly less than CIN2, and you see the undifferentiated basal cells in the lower 2/3, but the rest of it is still differentiating, but it is differentiating abnormally. And I will show you this, okay, in a minute. So the answer is graded disorganization and differentiation, which if you look at high power here, can you see? You can see this in the lower part of the epithelium. You see it's like basal cells that are abnormal and it looks blue. And that's why people say, lower 1/3. There's nothing lower 1/3 about it. The whole thing above that is also showing disorganization, albeit there is some degree, a good degree actually, of differentiation in CIN1, but not more in CIN2. Is there anything? There's more in, less in CIN2 and absolutely nothing in CIN3. All right. Anybody in the chat box has any questions on this?
Questions, nahi, ma'am. There are comments that even NC ratio and increase in mitosis is seen in SIN 1, 2, and 3.
Of course it is. It's bound to be, but it is in better differentiated cells. It's not in basal and parabasal cells. You recognize them as being high intermediates or low intermediates, as the case may be. Of course it is going to be, because you have disorganization. You have lacking differentiation. Okay, so that is how you are able to do it. And you see this beautiful differentiation over here. All right? Lack of differentiation, which you should only see in the basal layer, is now occupying this the lower 2/3. But above that, there is differentiation. There is increased nucleus cytoplasmic ratio. But in what? Not in basals and parabasals. It's the progeny of these basals and parabasals which are differentiating. Okay, so just like what you would see in a well-differentiated squamous carcinoma, right? You will see that there are pearls and other things being formed. In a moderately differentiated squamous carcinoma, less pearls, etc. are being formed. Keratinization is less. And in a very poorly differentiated carcinoma, you may not see any keratinization at all. And that is exactly what is happening, except that this is in an in situ situation. That's in situ, this is in situ, that's in situ. It is not a carcinoma in situ, which only CIN3 is, but all of this is neoplasia in the epithelium. It's intraepithelial neoplasia. Have you understood this? And if you haven't, we have the next chance in the neoplasia number 2 to go through this. All right, so now we come to the hallmarks of cancer. I'm not terribly excited about hallmarks of cancer because it's not something that you need to understand in great detail. What I just told you before is what you need to understand. Once you know that something is a cancer, yes, there is sustained proliferation. It evades growth suppressors, otherwise it may not grow. So it could be stimulation of growth. It could be evading growth suppressors, because growth is one of the defining features in the definition that we have of cancer, right? There is invasion and metastasis, which we have talked about. Cancers try to attain immortality, and therefore, see these beautiful symbols? You see this infinity, the symbol for infinity. So enabling replicative immortality, angiogenesis, you know about this. Resisting cell death, that means apoptosis doesn't work well in that system because the tumor has learned how to avoid apoptosis. Okay, so these were the 6 hallmarks that the high priests of tumor biology, that's Douglas Hanahan and Robert Weinberg, they published in 2002. And it still is being reproduced in our textbooks, okay? They added some, 10 years later, they added 4 other or hallmarks, okay? 2 are of the cancer cell, meaning that they deregulate cellular energetics. Then you have, they avoid immune destruction. So now you know that there's a lot of therapeutics that tries to prevent this, the immune destruction that the tumor cells are the killer cells are trying to get rid of the tumor cells, but the tumor cells have their defenses against the cytotoxic T cells. Okay, so that's how they avoid immune destruction. Then there is the stroma or the microenvironment, which promotes inflammation, and the cytokines in that inflammation provide growth factor stimulation to the tumor. You have genomic instability because a number of tumors actually show you, they are very unstable and they're constantly undergoing problems in the genome, either whole chromosomes, or there is chromosomal instability, or there is genetic instability in which portions of the genome are continuously being broken down and repair is not taking place. All right. So this is the whole thing, which I just said now. I've put all of this. This is, of course, taken from, it's an iconic picture that Robbins has over here, but I have modified it. Okay. Anybody wishes to take some kind of screenshots, you may do it. And I love the pictures because they've managed to show very well by symbols everything that we talked about now. All right. I just want to tell you in short something about autophagy and something about anoikis, because these are words that are in many textbooks and people don't understand them. The Warburg effect is about what they mean by cellular energetics is our normal respiration that in normal cells is through the mitochondria, okay, which is oxidative phosphorylation. But what happens in many tumors is that is bypassed, and tumors use what is called the glycolytic pathway. Now that is a very inefficient form of energy. It is also requires a huge amount of glucose that has to be taken up by the tumor because its energy consumption is around glucose. So you should not feel surprised that somebody who has a tumor, or for that matter, somebody who's diabetic, starts to lose weight because the tumor is literally catabolizing all the sources of glucose that are present, that's cancers by and large, I'm talking about, that are present in the stores. So that is what is called the Warburg effect. Now, any one of you can tell me where in our diagnostic armamentarium, and I don't only mean pathology, we use this Warburg effect to
PET scan.
PET scan. PET CT.
in what? In a PET scan?
Yes, ma'am.
In a PET scan. What PET scan? What PET scan are we talking about? Huh? Aishwarya? What is this? What is a PET scan? What is PET? Positron emission tomography. So what kind of PET scans can you have? It's a metabolic scan. Yeah. So by itself, it tells you nothing. It is when the PET scan is superimposed on a CT that you know what is actually happening and what is being metabolized. So what is being metabolized? What is that PET scan? Oh, there are types and types of PET scan, all right? The common one that is done is known as FDG PET. So what is FDG PET?
Fluorodeoxyglucose.
Yes. So what is done in that? Who answered that question? I don't see all your faces here, so I don't know. Who answered the question? It was a big voice. Huh?
Anees, ma'am.
It's us. Sikandar. Who?
Yes, ma'am.
You answer the question, Sikandar?
By my side, yeah, fluorodeoxyglucose, ma'am.
Okay, very good. But what do you do in a PET scan?
Ma'am, we inject the radioactive glucose.
Absolutely, that's right. And you inject that, and because any tissues that are avidly taking up glucose, yeah, normal tissues that avidly take up glucose are the brain, because the brain is very greedy for glucose, and that's why you feel lightheaded if you don't have glucose in your system, right? If you're hypoglycemic, you start to faint, okay, because the brain needs a lot of glucose. So normally, yes, there are tissues that take up a lot of glucose, but apart from that, it's inflammation and it is tumors, okay? So tuberculosis will take up FDG PET, very inflamed tissues, or if a child is crying and its thoracic muscles are going to take up a lot of glucose, because when you're using your muscles take up glucose, you know that, no? An athlete who's running, etc., and when it's happening, if you're just at rest, your muscles are not taking up glucose, but if you're using them, then you overuse them, then you get lactic acid that comes out of this glycolytic pathway. All right, very good. So let's quickly go through this. Yeah. Hallmark number 1 is proliferation, and that is regulated by growth factors, cell surface receptors for those growth factors, and downstream signaling through kinases, all right? And all of these, the growth factors, the proteins that we call growth factors, the proteins that we call cell surface receptors, and the kinases. And please remember, these are all proteins, okay? Are coded for by these genes, which are called oncogenes, because they happen to be involved in tumor production and tumor progression, okay? You also have paracrine mechanisms. What is paracrine means? That the cell secretes growth factors, which then act on the cell itself. So it is a continuous process of a self-stimulation by paracrine mechanisms, or by cells that are close by to the cell. So there you can have those kind of mechanisms. You have actimate. How do these growth factors, whether they happen to be cell surface receptors, kinases, or actual growth factors, they often have mutations in the genes, which are known as activating mutations, okay? And there is this very interesting phenomenon that under normal circumstances, if something is proliferating a lot, then, the cell actually pushes this, the mechanisms within the cell push the cell into, death, that is by apoptosis or senescence. This is, and yet, tumors have learned how to evade this process. Now, all of you should have heard of receptor tyrosine kinases. They are very, very important now. What are they? They are tyrosine, they have a tyrosine residue that is within the cell. They have an extra cellular kind of structure, which when there is a ligand, the ligand is something that comes, it's like a key that gets into the lock. When a ligand comes to the cell membrane, these receptors dimerize. That means they are sitting by themselves, they come together. And like I will show you in the next particular slide, it's like how cricketers on the field or fielders are taught how to hop a ball. No, they are supposed to put their hands together and open their palms, and the ball is supposed to float into their hands. And that is exactly what our receptors do. So when this tyrosine is phosphorylated, this transfers the phosphorus molecule to its substrate, and the substrate then, with downstream signaling, gives rise to a cellular response. And you should be familiar with these tyrosine kinase receptors. Can any one of you tell me the names of any of these tyrosine kinase receptors? They're known as
IGFR.
EGFR, excellent. Anything else?
Yes,
all common things that we know. EGFR, KIT, PDGFR. Yes, very good. RET, which is involved in what? RET involved in medullary carcinoma, thyroid, yes, and
L2.
Yes, and also in papillary carcinoma of the thyroid as well as in MEN2. Then you have VGFR, all right, which is involved in renal cell tumors. You have FGF and all of these. All receptor tyrosine kinases, and they are also considered as part of this whole growth factor system, which in this case we call oncogenes. Okay, so all of these are listed as oncogenes. Okay, sorry, annotation. I'm going to decline it. Right. So here is one very important receptor which we look at all the time. It's important. It's there in lung cancer and so many other cancers now that we are finding out. So you see this? This is the way that the ligand causes dimerization of the receptor. And I am always taken by this analogy. It's like how people hop a ball. You know, the fielders do it in cricket. And then there is a downstream signaling pathway that you can do. This is the MAP kinase pathway in this case. And that signal ultimately leads to several effects. It leads to angiogenesis in some situations. It leads to growth in some situations. And it also leads to cell motility. And through cell motility, you get invasion and then metastasis. Okay, so these are the growth factor receptors. Okay, now the hallmark 2 is evading growth suppressors, and I don't need to tell you about this. There's all about tumor suppressor genes, and the ones that we talk about most is RB and T. P53 RB is the retinoblastoma gene, which codes for the retinoblastoma protein, and the TP53 codes for the P53. Now, these are known as gatekeepers. Nowadays, your Robbins talks about it as governors, whatever it is, but it governs the cell cycle, you know, whether cells are allowed to enter the cell cycle or they are allowed to exit the cell cycle. That's what these growth tumor suppressor genes are all about. And I've just listed some of them over here. And how do you lose these tumor suppressor gene functions? You lose it through mutations, deletions, or something that silences these. The gene is perfectly normal, okay, but it is unable to be transcribed, and that may be because its promoter has been silenced by methylation or maybe even by histone acetylization or deacetylization, as the case may be. Okay, so that's what it is all about. Now you know this, and I'm not going to go into any details, but these are all the different rare syndromes. They are by no means common, and everybody knows about the Knudsen's hypothesis. I don't have the time to go into it, but if one of the alleles is inherited and it's abnormal, if during the life of this individual, you have both alleles are, the other allele is lost or it is mutated, then you know, the retinoblastoma happens. Now, I want you to think about this. I'm not going to ask you to answer about it now, but isn't it strange that say you have, in a family, there is the retinoblastoma gene anomaly, and every cell in the body has that abnormal gene. And yet, it's only retinoblastoma that occurs. Why do we not get squamous carcinomas? Why don't we get other tumors, maybe in the colon, in the gut, in the soft tissues, or anything? Okay, we do get it in the Lee-Fraumeni syndrome when you have P53 is abnormal in a family. Yes, you get breast cancer, brain tumors, acute leukemias, bone soft tissue sarcomas, adrenocortical carcinomas. You also get endometrial carcinomas and so many other carcinomas. But why does it not happen with the retinoblastoma gene? So I think that's something that you have to think about. Then resisting apoptosis, and you know that apoptosis is a response to a variety of developmental, physiological, and pathological needs. Do you know how you get your fingers, how fingers are formed in your hand? If you know what a hand looked like in embryological life, it's like this. It's like a fist. Nothing. It's just a blob of skin with its subcutaneous tissue and developing cartilage in that. And then what happens? This apoptosis causes loss of the in-between tissues, and fingers begin to form. So apoptosis is very, very important in developmental physiological conditions. It's also very important in pathological needs because you don't want damaged cells to hang around, and apoptosis is responsible for making those damaged cells die. Okay, so obviously, it is an important mechanism in the maintenance of cell and tissue integrity. Now think, if cell and tissue integrity cannot be maintained, okay, because apoptosis is abrogated, imagine you're going to get exactly what we described in cancer. What is that? Anvesha, what is it? Disorganization of tissue. Okay, it's for the maintenance of cell and tissue integrity, you require cells in different situations, different cells, to be able to either become senescent or to die. All right. So the main triggers are DNA damage and excessive proliferation. That's what I told you. If some things are going out of hand, then apoptosis will be triggered. And it's balanced with pro and anti-apoptotic factors. Now, this I have taken from Robbins, and I want you to, I don't want you to even look at these pictures. This is about P53, that is about RB. And I want you to do some homework, because at the end of this talk, which we will come to in the next 15 minutes, I am going to give you some homework. So please look at these pictures. They're there in the Robbins. All of you, I think, have Robbins either at your homes or in the libraries, because you are going to have to answer some questions based on what you are seeing over here. Okay, and one of the questions is right here. The RB question you will answer as homework, but here is something. Can somebody? This is an endometrium on the left hand side, and it's an endometrial carcinoma. And I've said that it is high grade and it's probably serous. Can anybody tell me what this brown staining is over there? I think it's easier if we start with the right hand side. Yes, you see this brown stain over here? This is follicular lymphoma grade 1 or 2, and this is follicular hyperplasia. So what's this brown stain? Yes, somebody said it. What?
BCL2.
BCL2. Yes, wonderful. That's BCL2. So now tell me, what is the brown stain on the left hand?
P53.
P53. Excellent. Whoever said it? Who said it?
Anise.
Anise. Yes, it's P53. And Anise, now you will have to answer the question. We just said that P53 has to be removed, is removed from a system, and then these tumors are unable to go into apoptosis, right? So we should not see P53, right, Anise? So we're seeing huge amounts of P53. It should not be there. We are seeing huge amounts. And we see this all the time, huge amounts of P53.
Yes,
and when we see progression, yes, Anise.
We said the mutated P53.
Yes, I know it is mutated P53, but I don't think I have an antibody to mutated P53. I just have an antibody to P53 protein.
Yes, ma'am.
Oh, so why am I seeing
Overexpression.
Overexpression? Do you think that this is just overexpression?
No.
No. Okay. So here is another question, and I'll ask you.
We say it is low.
Yes, Anise? You
Loss of
wanted to say
P53
something?
mutation. Loss of P53 mutation.
Yeah, I know there's a P53 mutation, and P53 mutation is not allowing this, the apoptosis to occur. Okay, it is there it is mutant. The protein is mutant, but I am showing P53, and there are many mutations in P53, but I'm using an antibody to P53. So why? All right, now you think about it and come back. We will do homework next time, okay? So you've got P53 as well that you have to do homework. Now you know about telomerase, that all cells start to, what do? The telomeres shorten as the cell differentiates and becomes senescent, okay? And that's known as Hayflick's principle, right? So there's only some 51 or 52 cycles of growth, that means mitosis, that can actually happen. And this happens because the telomeres are lost. So what happens if telomeres are lost? Yes, we know that the cell does not proliferate, but why? Why? Anybody can tell me? So if you don't know that, then that's another question. Is there anybody who's saying it in the chat box about P53 and the telomeres? Why we stop proliferating if the telomeres are not there?
Telomeres are responsible for aging effect, and P53 is not degraded.
Yeah, so whoever said that P53 is not degraded is absolutely right. Okay, P53 is normally degraded very quickly by the ubiquitin pathway. Right? Mutant P53 protein cannot be degraded. And that's the reason it hangs around in the tissues. But it is useless. It cannot actually function. So there's plenty of P53, mutant P53, and it cannot be degraded, and it doesn't work. Okay, so it has no, the function is completely lost, but it hangs around. So it's like having a useless body over there that's not able to do its function. Very good. But somebody has not answered the question as to why we need telomeres to actually proliferate. In the first place, why do we need telomeres? Yes,
Somebody has answered. It's Hayflick limit.
Yes, I know that. But why is there a Hayflick limit? Why can't we go proliferating forever? Hayflick just found out the limit. Okay, and then we realized that it is because of telomeres. But why? What do telomeres actually do? What is their function? So now somebody will have to tell me the next time. And I'm not going to go into how it happens. You have this there is a whole, telomerase, this whole protein has a reverse transcriptase, that is telomerase, reverse transcriptase, etc. It's in a complex which rejuvenates telomeres, and that's what cancer cells are able to do. Okay, and you see the Nobel Prize that was won. All right, then we have neoangiogenesis. And neoangiogenesis is a marvelous thing, that tumor cells promote neoangiogenesis because they secrete VEGF, okay? They also have VEGF receptors, so it is almost like an auto stimulation of themselves. And we know that tumors fail to grow larger than 2 millimeters if neoangiogenesis is absent. Okay. Now, at some stage, we were very interested in this in Tata Hospital. Okay, because I used to do head and neck cancers, and we see this problem a lot of the time. A huge lymph node. Okay, but you can't find the primary. This is very common. It is so small that you cannot detect it, not even with PET scans. So the question that we asked ourselves is, why does a tumor grow inadequately at the primary site, yet it grows profusely at the metastatic site? Okay? And we asked this question and we answered it. And we answered it by trying to, with a hypothesis. And our hypothesis was that these small tumors, which we can't see, okay, the primary tumors, are angiogenically incompetent. So they are unable to make blood vessels, and therefore they undergo cycles of growth and cell death and growth and cell death, and never grow to larger than maybe 2 millimeters. Okay, so we took the large lymph node because we couldn't take the primary tumor, and we compared them to those with known primaries, and we found that these metastases from unknown primaries actually had relative angiogenic incompetence. But I'm sure, I hope that some of you are thinking, hey, if they're angiogenically incompetent, then why are they growing in the lymph node? Well, we had done some work before, and that showed that angiogenesis is redundant for tumor growth in the lymph nodes. Why? Because the tumors at the lymph nodes are so full of blood vessels that they don't require neoangiogenesis. Okay, so this was some work that we did a long time ago, and it's important because if you are going to use anti-angiogenic therapy, and a lot of anti-angiogenic therapy exists, you cannot use it in the setting of this unknown primary situation, because those tumors have switched off their angiogenesis. First of all, they are angiogenically incompetent, and even normal, when I say normal, regular tumors that have metastasized to lymph nodes switch off their angiogenesis because the lymph nodes afford them a huge vascular and lymphatic supply which nourishes those tumors. Okay, I just thought this was important because this is not just in the air. Everything has some bearing on our clinical practice. Then, of course, we have invasion and metastasis, and you will see all of the steps. First of all, the tumor has to break its basement membrane. Then it has to enter into the tissues, that is through the extracellular matrix. It has to acquire motility. So epithelial cells which cannot move, they are fixed. Now they begin to get motility. Okay? And that is what is called EMT, epithelial to mesenchymal transformation. What? Why? Because epithelial cells have to acquire the traits of mesenchymal cells, because mesenchymal cells like fibroblasts, etc., have the ability to move. That's what they do. So also do lymphocytes. So also do neutrophils. All right, all of them move. So this is what is the first thing, or during this invasion process, the epithelial cells acquire epithelial to mesenchymal transformation or transgression. Okay. And the other thing that is important, and we'll talk about it in a little while, is anoikis. Okay. Now, how do these tumors actually, what do they lose? They gain all of this invasiveness, motility, all of that, but they lose something. They lose E-cadherin, they lose other adhesion molecules that make them stick together. And this is known as loss of contact inhibition. Okay, so it's the modification of adhesion molecules, and E-cadherin is a very important adhesion molecule, and all of you pathologists know about E-cadherin because it is characteristically lost in some carcinomas. And here is an in situ lobular carcinoma. Okay, it's associated with an intraductal carcinoma over here, which happens. And in this case, you see beautifully that the intraductal carcinoma has not lost E-cadherin expression. You can see the brown membrane because that's where E-cadherin is on the cell membrane. And here, these cells have lost their E-cadherin. Okay, we use it in breast cancer diagnosis. We use it in sometimes some stomach cancer diagnosis, etc. And this is how the metastatic cascade actually happens. You can look it up in your books. I don't think it's important. I don't think it's important for me to talk about it, but there's angiogenesis, motility, intravasation into the blood vessels, then survival in the vasculature, and at every spot, the tumor cells are vulnerable. And only those cells which are able to survive this journey actually reach and come, are able to get out of the blood vessels and get to the metastatic site. Okay, and all cells in a tumor may not be capable of metastasis. So the metastatic phenotype may develop over a process by clonal heterogeneity. So it starts without being metastatic. Then suddenly you get a cell which may have acquired something, the invasion, the ability to break down barriers, etc. Then it grows there, and then it may actually metastasize, and the metastasis may show you even more clonal heterogeneity, while the main tumor, yes, still maintains its metastatic phenotype and gets other phenotypes, because there's constant, we talked about genetic instability, and more different kinds of clones are produced in the tumor. Okay, now this is the molecular pathology of cancer. We use this in diagnosis. We look for mutations, we look for deletions, we look for amplification in HER2 and NMIC. These are amplification of various genes which are not, we look for loss of not only these, but many other genes that we look for. We look for gene rearrangements, which are common in sarcomas, lymphomas, and in leukemias. And we also look for the final end product. Is there any protein that is just like we did in P53, all right? We know it's mutated P53, otherwise it would have been degraded, okay? So by looking at the protein, we will know what has actually happened in the genome, whether there is a mutation or there is an epigenetic modification, etc. So this is, in short, the molecular pathology of cancer. Okay, I have just a little bit, before I give you the homework, I just want to tell you quickly about autophagy and anointis, okay? And this is something that I feel is not touched upon enough. And this is about housekeeping and homelessness in cancer. Now, I'll tell you why we say housekeeping. There was a Nobel Prize that was awarded to this scientist, Yoshinori Oshumi, who made the breakthrough when studying yeast, that bakers used in whether it is bread or cake or whatever. And he discovered that there are these, and of course, he did work on something that had been done by Christian Duve, but what did he discover? That autophagy takes place in all kinds of cells, and a yeast is a fungal cell, okay? It means self-eating. And what is this? All cells with nuclei have, that's what eukaryotic cells are, have this capability. And what it does is that the cell sequesters, that means it collects within a double-layered membranous vesicle. It's called the autophagosome. We've heard of lysosomes and phagosomes, but here, this is a phagosome which is eating up parts of the cell. It's not phagocytosing extraneous material. And that autophagosome, it is then delivered into the lysosome for degradation. Now you might ask, why? Why do we need this kind of autophagy? All right, that's what it does. It removes long-lived proteins, damaged or obsolete organelles, okay? Toxic proteins. If phagocytosis is inefficient, then the autophagosome is formed, and it has some extremely potent enzymes which it delivers to the lysosome, and it therefore plays a very important part in infection, aging, degenerative disease, and cancer. All right, if you lack autophagy, you are various degenerative diseases have defective autophagy. So your cells are in effect so messy that they trigger apoptosis and they die. Okay. So housekeeping this is a kind of tumor housekeeping, not tumor, it's just cellular housekeeping. It provides material for metabolic reactions, repair, because it just recycles them. Okay? Proteases break down, hydrolases break down fat, sugars, nucleic acids, okay? So they clear all this stuff. And autophagy keeps cell processes intact and thereby enhances cell survival. Now, we don't want cell survival in cancer. It should, these cells should be pushed into apoptosis. But if the cancer cell is able to evade autophagy, then apoptosis will not occur. Okay, so that's why autophagy is a very important part of what's going on in cancer. It makes tumors resistant to chemotherapy and radiation therapy, which should actually kill cells. But because autophagy is sometimes overactive in cancer, it's promoting cell survival. It is also clearing up all the debris that chemotherapy and radiation therapy, produce, and tumor dormancy is also, so it helps in tumor progression and metastasis. Now, what is anoikis? This was first, discovered years and years ago in 1994, okay? And anoikis really means homelessness. And I showed you this diagram. When a cell loses its adhesion, epithelial cells in particular, it can happen in endothelial cells, can happen in mesothelial cells. When they lose their adhesion to their extracellular matrix, they should undergo anoikis. Okay, why? Because the moment that happens, that cell is not supposed to be where it is, and therefore it should die. That is how nature prevents cells that are not supposed to be motile from going where they are not supposed to be. And so it triggers apoptosis. Okay? There are a whole lot of ways in which this is regulated. Much of it is regulated through integrins. Okay? And here it is. If the cell is not attached to the extracellular matrix, you get apoptosis. Now, there are a whole lot of physiological reasons for why anoikis does not happen. Okay, cells like lymphocytes, neutrophils are very resistant to anoikis because they have to be motile. They have to move from one place to another. Also, if fibroblasts, etc., are healing, if there is wound healing, then of course you cannot have anoikis. Otherwise, how will the fibroblasts go to the different parts of this healing wound and actually begin to bring the wound together? So they are also spared from anoikis. But unfortunately, invading cancer cells survive as they have acquired resistance to apoptosis, and anoikis does not work in this. And that's, again, the whole story of epithelial to mesenchymal transition. And this phenomenon occurs naturally. It is activated during wound healing, inflammation, or embryogenesis. Imagine, in embryos, cells have to move. They go from one part to another. And if there was anoikis, I think that we would probably not be able to form the organs that we are supposed to form. Okay, so I think it's so beautiful. All right, autophagy and anoikis are the yin and yang. Do you know what is yin and yang? Gurtej? You know what is yin and yang? Yin and yang is the two complementary parts of which in Chinese philosophy. Okay, if you have a good, you have a bad. If you in every system, the Chinese believe you have, if there's death, there is life, and so on and so forth. So autophagy and anoikis are the yin and yang, because autophagy promotes survival, anoikis promotes death, of the struggle between life and death. Okay, each of us, think about it, has within ourselves the decision-making machinery to either live or die. And this is a complex interplay of factors that ultimately determines the fate of each cell. The decision is based on the internal state of the cell and that of its microenvironment. And you know this, no? Immortality is a dream that cancer cells try to achieve. That's what cancer is a lot about. Okay. So, and I just put this up because it's a beautiful poem written all the way in the 16th century. Okay, he said, "No man is an island, entire of itself. Every man is a piece of the continent, a part of the main," meaning the mainland. So, think about this, and the next time we will talk a lot about this, about cells not being just of themselves. They are part of a whole system, of a tissue, and the tissue is part of the body. And all of this, what does it have to do with cancer? All right, so now we have homework. All right, and here is the homework. The homework is, you may not answer the first question. I will answer it for you, okay, the next time. Why do, see, if RB and TP53 are so important in cells, and they, in the whole cell system, when they developed RB and TP53 null mice, that means they had no RB and no TP53, they developed normally. So, going on, okay? Why are only a few organs targets of cancer in inherited syndromes? Okay, in APC, you get it only in the gut. Okay, and yes, if you have another form of APC, then you get Gardner syndrome, etc. But if you only inherit a particular form of this APC gene, why is only the gut involved? Huh? So take down these the homework. Why is P16? And we do it all the time in our labs, a surrogate marker for HPV in some systems. Okay, so P16 is a surrogate marker. It's like we use P53 in the lab, and to show the P53 protein. We also use immunohistochemistry to see P16. Okay, and the last question is, what is the pathogenesis of squamous carcinoma? Okay, just think about these things. I think there was one more that I thought that you guys should know, and what was that? That was, yes?
Role of telomerase, ma'am.
Ah, yes, about telomerase. So you'll have to remind me about that. I will write it in the next thing. Okay. So this is some of the homework you are expected to do, and we will take the homework the next time that we start. So we'll have 10 minutes of homework, and then we will do neoplasia part 2. So I'll say